Myself, shirisha. I am from Government Degree College Autonomous. Now I am telling about the Griffith's experiment from molecular biology. In this content, there are 6 steps: initial discoveries, Griffith's experiment and transformation principles, experiments, conclusions, DNA and genetic materials. Initial discoveries. The search for genetic material started during the mid-19th century. The principle of inheritance was discovered by Mendel. The principle of inheritance was discovered by Mendel. Even though the chromosomal components were identified, the material which is responsible for inheritance remained unanswered. It took a long time for the acceptance of DNA as the genetic transformation. Griffith's experiment. Griffith's experiment was a stepping stone for the discovery of genetic material. Frederick Griffith's experiments were conducted with Streptococcus pneumonia. During the experiment, Griffiths cultured Streptococcus pneumonia bacteria which showed two patterns of growth. One culture plate consists of smooth shine colonies while other consists of rough colonies. The differences was due to the presence of mucus coat in S-strain bacteria, whereas the R-strain bacteria lacked them. Experiment step 1. Griffith injected both S and R strains to mice. The one which was infected with the S strain developed pneumonia and died, while that infected with the R strain stayed alive. Step 2. Griffith heat killed the S strain bacteria and injected to the mice, but the mice stayed alive. Then he mixed the heat-killed S and live R strains. This mixture was injected into mice and they died. He found living S strain bacteria in the dead mice. Conclusion. Based on the observation, Griffiths concluded that R strain bacteria had been transformed by the heat-killed S strain bacteria. The R strain inherited some transforming principle from the heat-killed S strain bacteria and had enabled the R strain to synthesize a smooth polysaccharide coat which made them virulent. He assumed this transforming principle as genetic material. DNA and genetic material. Griffith's experiment was a turning point towards the discovery of hereditary material. However, it failed to explain the biochemistry of genetic material. Hence, a group of scientists, Oswald Avery, Colin MacLeod, and Maclean continued the Griffith experiment in search of biochemical nature of the hereditary material. Their discovery reversed the concept of protein as genetic material to DNA. Avery, MacLeod, and M.C. Critt's conclusion. They extended and purified protein, DNA, RNA, and other biomolecules from the heat-killed S strain bacteria. They discovered that DNA is the genetic material and it is alone responsible for the transformation of the R strain bacteria. They observed that protein digesting enzyme proteinase and RNA digesting enzyme RNase did not inhibit transformation, but DNase did. Although it was not acceptable by all, they concluded DNA has genetic material. Thank you for watching and listening this video.